Right, let me get a PowerPoint. So some of this you might have encountered during some of the exercise over the past 3 days through Go terms and transforming things into orthologs. So I'm basically gonna wind back a little bit and explain a little bit more about what orthology and ontology actually mean and how you can actually use them. Why they're important to kind of take into account when you're doing your analyses and things. The main thing for these these are basically just methods of categorizing your genes as you go. So everything in biology that makes it useful is essentially a category. You want to know what a thing is and what it might do and what it might be related to in order for it to be useful to you. So there are a couple of main ways that we categorize things just in general in life, not just within genomics. The main thing is through taxonomy. So this is a hierarchical way of organizing things in life. You start with one big category at the top, but descend down. This is all relatively simple things, but it's hierarchical. You don't have links in between all the lower categories within the taxonomy. Ontology, so like gene ontology, is different in that it is more like a big network of boxes that you have put stuff into, and there are lots of little connections between all of those boxes. And even in this, you can mark sort of partial relations as well. So there can be direct relationships and partial relationships within the big network of the ontology. Ontology is just the formal description or categorization of a thing. It is not restricted to biology, like I just said a minute ago. It could be used in mathematics or physics or sociology. But we're interested in it in biology because we can use it to make more sense of what genes might do or what they might be associated with. So we have 3 main ontologies that we look at. There's molecular function, cellular components, and biological process. So molecular functions are at the molecular level, and can be things like toxin activity or enzyme activity. And then you have the cellular component, so the localization of where it performs, whether that's cilia or mitochondria or other organelles. And then you have the full biological processes as well, so like pyrimidine biosynthesis here. We like ontology because it gives us boxes to put things in, and also because it allows us to look at and compare genes across many, probably quite widely dispersed organisms that might all share a similar function. So here we have the example of cyanell, chloroplast, and the apicoplast. Two of these 3 things are quite obviously photosynthetic things. They are green and they look nice under a microscope, and they photosynthesize. And then we have the apicoplast, which is here also colored green, but it's not green in real life. It is non-photosynthetic. It's found in things like toxoplasma. But this will share genes with the other 2 photosynthetic organs. And you can basically look across this Go term and you can look across all these different organisms and compare what they might or might not be doing and why they are different. And one thing, to start doing that, you can do something called gene enrichment. You can work out if your set of genes might have an overrepresentation of a specific Go term compared to the rest of the genome, and whether or not that is statistically significant or not. So to do this, I'm going to very briefly go over how these sort of things are done. These are not, you don't necessarily need to remember. It's just sometimes it's nice to know what goes on behind things. But to calculate enrichment, we use something called a one-tailed Fisher's exact test. And usually this is used for contingency tables in small sample sizes, so comparing little groups of things in sort of like a grid fashion, and seeing whether there's any statistical differences between them. In a gene versus a whole genome situation, that is not a small data set. It is quite large, and that increases your risk of false positives. So we do something, so we correct for that using a couple of different methods, Bonferroni and then the Benjamini test as well. And we do these to just to double check and make sure that the p-values that have been calculated are actually reliable in the first place. So yeah, the main thing that you need to take from this is when you're calculating Go enrichment, you have your set of genes in your study. They all have associated Go terms within them. And then you have your main population also with all of their terms that are associated with them. And you're comparing those two to see if there's a higher probability of getting a particular Go term in your little study set in comparison to the rest of the population that you're looking at. And when you do that on our sites, that will give you this kind of table. And the columns that you're looking at are these ones at the end. You're looking at the p-value to make sure that's significant, and you're also checking that these two are also reasonable as well. So yeah, that is the basics of Go enrichment. Are there any questions from that before I move on to orthology? I'll take that.
Is there a cutoff number below which this type of enrichment is meaningless?
So it's kind of like any p-value score. The smaller it is, the better. You want it to be below 0.05.
No, what I meant is that it's different to have enrichment with 100 genes and different to have enrichment with 100,000 genes.
Not really.
Not really.
Unfortunately, there's not. Well, there isn't a different way to go about testing it. You're basically just looking for it being significant. And the other thing, these categorization things, you use them as guidelines rather than definite things. Like you wouldn't just look at this and go, oh, this is enrichment, so this particular bit of metabolism is enriched, therefore it must be important and significant for that organism. You'd want to take that and actually go test and see if it is in the first place. You don't want to just take these at face value necessarily.
Thank you.
Can I just interject something here as well, Helen? Is that okay?
Go for it.
So it's for the numbers you were talking about, it's fine, but where you want to be really careful is where there's only one gene in your list. Those do sometimes come out as significant. So if you only have one gene with, I don't know, let's pick one of the names of this one with a serine endopeptidase activity, and that is in your list, it may come out as significant, but because there's only an N of 1, you really don't know if that's actually random that it's in your list or not. So those ones where you have something that looks significant, but actually the fourth column in this, the genes in your result with this term, if the value in that is very low, you do want to be a bit wary, even if the p-value is good.
Yeah, that is another valid thing. Thank you. Okay, cool. So on to orthology. So orthology, again, is another way of categorizing your genes and what they do. Comes in a couple of different forms. The main ones that we'll be looking at today are orthologs and paralogs. So Orthologs are, it's just the study of how genes are related. It comes in two main categories, which is homologs and analogs. Homologs are shared, they have a shared origin, and analogs might share a function, but they don't share an origin. And then within homologs, we can split that into three more categories. We have orthologs, which all originate from a single ancestor, and then paralogs that occur via gene duplication, and then xenologs, which we don't really carry in ViewPathDB. We don't identify them. They are derived from horizontal gene transfers, so they'd be more relevant to bacteria and things, but it's just something to know. So here we have an example of how this kind of works. We have the histone gene. We start with histone gene here in mammals, and then the HNS gene that occurs in bacteria. These two things have vaguely similar functions in their organisms, but they occurred by different methods, and they occurred through convergent evolution. Within the so this makes these two things analogs. Within the histone genes, you have gene duplication events, which resulted in two different versions of this H1 histone gene. These will be your paralogs. And then you have a speciation event that results in these two different genes having different versions in different species, which are your orthologs. So H11 in humans and H11 in chimps are orthologs. And then the same for these two blue ones here. Orthology can be used to learn about your favorite genes. So this might be before you've decided to do anything practical in the lab, for instance, and you're trying to figure out what might be something interesting to look at or investigate further. You can learn about how conserved your gene is and how what their function might potentially be, and if there are any phylogenetic patterns that you see for your genes as well. So if we take this example, just kind of picked from the ToxoDB database, this is a hypothetical protein that we find in one of the strains of Toxoplasma gondii. And if we wanted to figure out what this thing is, what it might do, we can begin to look at orthology to give us a few different answers. So on ToxoDB, if you scroll down to the orthology and synteny group, you have a list of other genes that share the same orthology group, and you can look at the product description. And for this particular gene, you can begin to see that while there are a few sort of unspecified things, hypothetical proteins, you do get quite a few of the same or similar product descriptions, this serine threonine protein kinase 19. So this particular gene, we might start to think, oh, maybe it's this. Let's just make sure. We'll go and investigate the ortholog group a little bit further. So clicking on this link here will take you out to the OrthoMCL database, and it will give you a similar sort of summary page of that ortholog group and begin to describe what it is. So at the top of the page, you'll get this little summary. You see that there's keywords, so you can see which things occur the most in product description. So in this case, it is unknown, and then kinase and kinase 19. You're also given an EC number. An EC number, if you're not aware, are associated with specific metabolic pathways, and a 4-digit EC number shows that an exact function is defined. So for this group, we have this particular EC number function that is associated with 68 things in this ortholog group. This summary will also show you the PFAM domains. So you can sort of begin to get an idea of there's 180 of this PFAM domain and only one of this PFAM domain. So without even looking at the products and things, you can start to think this one is more likely to be the potential function if it has that as a function. The other thing from this little bit of the screen as well, you can download this whole ortholog group, so you can get the metadata from it. And probably the most useful thing you can get from it is the FASTA file, so you can get the FASTA protein sequence. So you get a sequence for each thing in the ortholog group and a little descriptive thing in the FASTA file, and you can take that away to your favorite alignment programs or tree making things, and perform your own phylogenetic analyses on it, if you so desire. But to begin with, before you do that, you can scroll down and you can begin to look at where this thing is found. And in this instant, we can see, so this only occurs in eukaryotes. It doesn't occur in bacteria at all. You can expand all of these little tabs as well. And if you do that, you can see that this particular gene, or this particular orthogroup, only occurs once in each species that it is in, well, most of the time. So it is very conserved. And then also within alveolates, which Toxoplasma gondii belongs to, it is restricted to coccidia, which could be an interesting thing to look at. It might not be, but it could be. So you can begin to see where it is distributed across the tree of life, things that it might be associated with that way. This page also will give you a list of proteins that it might be similar to. Here you can start to look at the descriptions again of what it's associated with, and again this protein kinase is popping up quite a lot. We actually have an inbuilt alignment tool here, which will also take you to ITOL Tree of Life tree visualizer program. And this will give you a very quick, dirty approach to defining the phylogeny of these things and having a quick peek at whether or not that side of things might be interested in. But I would recommend that if it is interesting and you want to take it and do other things with it, download those FASTA files and do your own alignments, make sure there isn't anything weird going on, and making sure your alignments are nice and clean before you make any real judgments. It'll also list further down the page PFAM domains. And again, I am a broken record at this point. More protein kinases. This is the group that it has 180 things in it. So taking all of that information together from the proteins and PFAM domains and the EC numbers and things, we can probably conclude that this gene that we were looking at in Toxoplasma is a serine threonine protein kinase. But you need to be quite careful when you're doing this, because it is sort of an extrapolation of evidence. Not everything in any database, whether it's ours or NCBI's or whatever, is necessarily going to be annotated correctly. So you need to take all the information that you have as a whole and make your own best judgment based on the available evidence as to whether you can trust it or not. So, for instance, if we go back here, if you have a whole bunch of different PFAM domains and there isn't quite as clear a difference in the counts of PFAMs, you would begin to trust that a little bit less than you would for this particular gene example. So this has all been based on the OrthoMCL website. OrthoMCL is dependent on the OrthoMCL algorithm, and this is essentially a massive reciprocal blast effort to identify what best hits occur between different proteins and lump them together in lots of different groups that are informative and useful to you as researchers. So basically what it does, it takes protein sequences from an organism of interest, blasts them all to try and against each other to try and find orthologs between species, and then paralogs within a species. It will then take all those numbers and put them into something called a similarity matrix, where it will begin to sort of find, well, it will find the similarities between all the things and provide this, which is then taken into something called the Markov clustering algorithm. Markov clustering algorithm, it basically works on networks, and it's driven by the idea of something called random walks. If you're not familiar with networks, networks are made up of things called nodes and edges. Nodes are these little dots here, so all of these dots are nodes, and in this case those would be your proteins that you are comparing. The edges are all the lines connecting the nodes in between. Those will be your similarity scores, in this case, connecting similar nodes. And random walks is basically when you go from one node to any other and you just take a route. And what MCL does, it will try and find clusters of nodes. So like up here, you can see one cluster and another down here, potentially. And in theory, those clusters regions will have many, many interconnecting little paths between them, and then far fewer connecting them to other clusters. MCL will then, it will sort of iterate over this, and it will begin to trim away edges that are less likely to be parts of these clusters. And with each turn of the algorithm, these will get fewer, and you will begin to get better defined clusters rather than one big spaghetti mess, which isn't really useful to anyone. One of these ortholog groups will it will look something like this. This is a very simplified view of it. But within an ortholog group, you will have your species. So here we've got a fly, a worm, and a yeast. They will have genes within them, which are all blasted against each other. Ones that have reciprocal best hits between all of them will sort of form the backbone of this being an ortholog group. And then within the species, you might have a couple of different genes, which will be best hits for each other, and those are your paralogs. In a larger example, so you would get something that looks like this. And these networks are visible from OrthoMCL right at the bottom of the page of any of the ortholog group pages. It will take you to one of these diagrams, and you will be able to see, you can adjust what the cutoff value is, so you can reduce the number of edges that are joining them, find like the most strongly connected ones, and you can also recolor all of these nodes by the EC numbers and the PFAM domain. So at the moment this is colored by taxa, which is why there's so many different ones. But yeah, EC number and PFAM domains will give you an idea of which clusters are associated with the different functions that might pop up in an ortholog group, if there are multiple ones in the first place. So OrthoMCL is comparing to over 800 species across the whole tree of life. This is made up of 150 core proteomes, which are curated ortholog groups. They sit basically as like the baseline level, and they don't change. They are sort of set. And then you have peripheral genomes that are mapped on top of those to add to the amount of data and things that we're comparing to. Just so you can make your comparisons in the ViewPathDB databases and take that information and try and find ortholog groups or transform your genes into those ortholog groups. So for the practical, which you'll be doing next, you will have a go at playing with the Go enrichment analyses on our websites, and also how to integrate and explore the OrthoMCL outputs into your searches that you're doing through the ViewPathDB websites. And you'll be looking at the phyletic pattern searches and the Orthology transformation tools in a little bit more detail than you have done over the last few days as well. So, any questions? I know that was quite quick. I think I spoke quite quickly, but
yes, if I may, I guess each ortholog group contains one gene per species, right?
I believe that depends on how many different strains of that species there are.
Okay, it's per strain as well. All right. Is
And
there
then
a
there's
way...
And
paralogs.
there's paralogs as well. Yes, thank you.
So, sorry, I didn't hear that. Each ortholog group would contain paralogs as well.
Yes.
Yes. So there can be many, many genes from a single species within an ortholog group. Some ortholog groups, you know, might have only one species in them. I think I've seen that on PlasmoDB.
Okay. Is there a way to, when you're looking two different species, for example, to map one-to-one orthologs? You know, because very often you can do that one-to-many or many-to-many, but let's say one-to-one orthologs.
You can get that from our parasite sites, if that's what you're asking. It's a little bit difficult to weed through all of the data on our sites, but if you use our strategy system and transform and then look at number of paralogs, I think sort by the paralog column in our result, you can get a good idea of one-to-one orthologs. Is that true, Omar? He might not be on, actually. I think he had to
Yeah,
jump
I think he
off
was dropped off. I'm going to speak for Ortho because I've been doing
Okay.
a lot of work on it. You can't always make a one-to-one connection. Sometimes one gene in one species is equally similar to two or more genes in another species. So almost all the time when you do a transform, you take a list of genes in one species and transform them to their orthologs, you'll get a larger number. You know, not every gene has multiple matches, but some genes do have multiple matches across to the other species. And sometimes if you really look at a particular comparison among a small group of genes, you can decide which is the closest match, which is the true reciprocal best hit, but not always. Oftentimes, you know, they're just, it's not possible. The two paralogs in the second species are very close to each other, and the ortholog in a third species is quite distant, and you just can't do anything except this one gene has two matches in this other species.
Okay, thank you.
So if you find an ortholog group, for example, in PlasmoDB, and you want to find orthologs in, say, GiardiaDB, then you would use the transform tool to try to find those?
So yes.
Yes. Or if you're looking at one ortholog group in a PlasmoDB species on the gene page, that you can click open a little button and it lists all of the orthologs in other PlasmoDB species.
Yeah, I was thinking about, you know, other parasites which are not Plasmodium.
You can so Stuart is right. If you're looking for orthologs within the taxonomy group that the site that are like PlasmoDB contains, then you can look at that table. But your original question was, say, you want to transform Plasmodium into Giardia, then you would use our little tools to take your Plasmodium list of genes over to ViewPathDB, where all of our organisms are represented, and then use the Transform by Orthology tool.
Or do it in OrthoMCL, which
What?
has its own. OrthoMCL also has its own website where you can do the transforms and the phyletic queries. You know, make a list of genes and find the orthologs that are in 2 specific species with a query.
Okay, thank you.
One last question from me. It's sometimes since I used OrthoDB. If I remember well, my question, by the way, is connected to one of the last slides, of which we've seen some synteny maps. Yes, precisely this. If I remember correctly, there are some orthogroup clusters that beyond a certain number of genes, one is unable to generate this particular type of cluster graph.
Yeah, we limit it to 499 genes, which is most of the orthogroups, but there are a few hundred orthogroups that have more genes than that, and the drawing tool just can't handle it.
Right. Is there a way, for example, if one can limit the number of species in the orthogroup to generate the maps, or to bring down this number to 400 and whatever?
We don't have a way of doing the cluster graph on a restricted number of species or genes in an orthogroup. Instead, we are going to build a simple way to draw a phylogenetic tree and use that instead of a cluster graph.
Thank you. Thank you, Ewan. That answers me.
I'm just curious, what questions would you be asking of this cluster graph? How would you be using it?
Well, the way I would be using it would be, for example, looking at the PFAM domains, EC numbers, trying to this way to sort out, or not sort out, trying to get some information regarding function.
So in other words, you'd be looking at subclusters and seeing if they have something in common?
Exactly, yeah.
Yeah, I think that will work okay on a phyllo tree as well.
Yeah.
Shall we go on to the exercise?
Yeah, that sounds great.